Hello and welcome to today's practical lesson. In this session, we'll be conducting a hands-on exploration of osmosis in action. We're about to head into our lab to explore a fascinating experiment that directly ties into the concepts we've been covering. But first, over to Sam, our lab technician, to outline what safety equipment we need.
Before we get started, make sure to put on your lab coat, safety goggles and gloves. Remember, safety is our top priority in any lab environment.
Thanks, Sam. While we're getting ready, let's quickly recap what we've learned so far. So far, we've covered the concept of osmosis, which is the movement of water molecules across a partially permeable membrane from an area of higher concentration to an area of lower concentration. We've also discussed how varying concentrations of solutions can influence the movement of water into and out of cells, thus affecting their mass and turgidity. Today, we'll observe this knowledge with a practical experiment. By placing potato cylinders in solutions of varying sucrose concentrations, we'll observe firsthand how osmosis affects the movement of water into and out of plant tissue. This experiment will not only reinforce your understanding of osmosis, but also provide you with the skills to analyze and interpret experimental data. We're almost ready to dive into the details of our practical. But first, let's explore why this experiment is both exciting and valuable. Osmosis is a critical concept in biology. It underpins many essential functions within living organisms. By conducting this experiment, you will gain knowledge of how osmosis works in plant cells, specifically in potato tissues. This deepens our comprehension of why water balance is vital for cell function and overall plant health. Understanding these processes helps you to appreciate why plants absorb water and nutrients from the soil, as well as why this is vital for their growth and survival. Investigating the effects of different solute concentrations on osmosis encourages you to think critically and solve problems systematically. You will need to consider why certain concentrations lead to changes in mass and length of the potato cylinders. This will develop your ability to identify patterns, propose explanations, and critically evaluate outcomes. This fosters a scientific mindset, where evidence-based reasoning helps you in understanding complex phenomena. This is also a skill that is valuable in everyday decision-making. On to the practical. Let's start by gathering our materials. Sam, what do we need?
So for this practical, we need potatoes for potato cylinders, a cork borer, a ruler, a top pan balance, sucrose solutions of varying concentrations, paper towels, and finally, a lab book to record our results.
Make sure your lab area is tidy and that all of your materials are within easy reach. We begin with our preparation stage. Firstly, we need to prepare a range of sugar solutions with varying concentrations. For instance, you might use distilled water without any sugar, followed by sugar solutions of 0.25, 0.5, 0.75, and 1.0 m concentration respectively. Label each boiling tube clearly to avoid any mix-up. Next, we need to prepare our potato cylinders using a cork borer to ensure that they are all of uniform size. Then, carefully trim each cylinder to the same length. We will use approximately 3 centimeters. After measuring their length, we need to weigh the potato cylinders. Use a digital balance to accurately measure the initial mass of each potato cylinder. We also need to ensure that we carefully record the precise weight of each potato cylinder in a table. Set up a table in your lab book as demonstrated. Then, fill in the first column with the concentrations of sucrose solution that you selected for this experiment. Next, assign a potato cylinder to each concentration and record the initial mass of each potato cylinder in the next column. Now it's time to start the experiment. Submerge each potato cylinder into a separate boiling tube that contains one of the sugar solutions. Keep the solutions labelled. Ensure that the potato cylinders are fully submerged and that each boiling tube has enough solution to be able to cover the potato completely. Leave the potato cylinders in the solutions for a set period, such as 1 hour. During this time, water will move in or out of the potato cells via osmosis. The direction of the movement will depend on the concentration of the sugar solution in question. After 1 hour, carefully remove each potato cylinder from its respective sugar solution. Gently blot your potato cylinders with a paper towel in order to remove any excess solution. Do not squeeze them, as this could alter their mass. Remeasure the mass of each potato cylinder using the digital balance. Record these final masses with as much precision as the initial measurements in your lab book. Your table should now look something like this. We can now use these measurements of the initial and final mass to conduct our analysis. In our analysis, we first want to calculate the change in mass that each potato cylinder has undergone. We do this by subtracting the initial mass from the final mass. After this, we can find the percentage change in mass for each potato cylinder. The general formula to find the percentage change in mass is the final mass minus the initial mass, divided by the initial mass multiplied by 100. For instance, if a potato cylinder had an initial mass of 5.34 grams and a final mass of 5.92 grams, then the percentage change would be 5.92 grams minus 5.34 grams divided by 5.34 times 100, which equals an increase of 10.9%. Enter your results into the table, and then calculate the rest of the percentage changes in the same way. Next, we need to draw a line graph to represent the percentage change in the masses of the potato cylinders. Place your sugar solution concentration on the x-axis and percentage change in mass on the y-axis. Take care to accurately plot each point, and then draw a line between each point. This visual representation helps us to identify the concentration at which there is no change in mass, which indicates an isotonic environment, where the concentration of solutes in the solution is equal to the concentration of solutes that are dissolved inside the potato cell cytoplasm. We should also analyse the graph to see where water moves into the cell by osmosis in a hypertonic solution. This is indicated by a gain in mass. Where water moves out of the cells by osmosis occurs in a hypertonic solution. This is shown on the graph by the areas that experience a loss of mass. Consider any surprising results or anomalies, and think of possible factors that might have influenced these, such as inconsistencies in cylinder size, varying exposure times, or inaccuracies in measurement, for example. By understanding and performing this experiment, we gain insights into how osmosis operates in plant cells, and the importance of solute concentration in maintaining cell turgor and overall plant health. This experiment not only reinforces the fundamental concept of osmosis, but also highlights the practical considerations that are necessary for accurate scientific investigations. That's the end of our lesson today. Let's recap. In today's lesson, we conducted a practical experiment to study the effects of solute concentration on osmosis in plant tissues, specifically using potato cylinders in various solutions. The key steps of this practical were preparing sugar solutions and potato cylinders, submerging the potato cylinders into the different sugar solutions, incubating them for a set period of time, and measuring our results. We then plotted the percentage mass changes and analysed our results. Our key takeaways for future practicals are to ensure accurate measurement and consistency in the size of biological samples, as well as to maintain precise control of experimental variables, such as time and concentration. This practical reinforced our understanding of osmosis and demonstrated the significance of concentration gradients in biological systems.
Watch the rest of the GCSE biology course at Sophos Education.